So the first step in getting the instructions out to the body is a process called transcription. And in transcription, the cell is going to make messenger, or mRNA. mRNA is just simply a copy of the DNA that can leave the cell. And remember from biology that RNA has A's and G's and C's, but instead of having T's, it has U, which stands for uracil. Okay, the first slide we have here is just a quick overview of what happens in transcription, how we make this mRNA. So the first step is showing that there is a double-stranded DNA molecule that once again gets opened up. In this case, the enzyme that opens it up is called RNA polymerase. So RNA polymerase, you can see on step 2, is creating another little bubble in DNA, but instead of copying both sides of the DNA, it's only copying one side, and in the process is making one molecule. That one molecule is in red here, and that molecule is the messenger RNA. So that's the basics of transcription. One side of the DNA gets copied, made into messenger RNA. Now let's look at a little bit closer up and get some of the details figured out. So here's our DNA molecule once again. I'm going to open it up first. Hopefully. It's a little tricky to grab here. There we go. Okay, again, the bases are already there. So I'm going to add some in just randomly. They're complementary because they belonged to the same DNA molecule. On the 3' end is where we're going to start transcription. And instead of putting down DNA, we're going to add RNA this time. It's going to go in this direction. C, G, C, C, T pairs with A pairs with U. So this is our messenger RNA that's being formed. Again, where do those bases or those nucleotides come from? They're out here in the nucleus, waiting to be paired up with the RNA polymerase. Now, a few more details about this process. The messenger RNA only transcribes one gene at a time. And in order to do that, it has to be able to know where the gene starts and ends. So one gene at a time. So we're going to talk about the starting to begin with. So the area on the DNA, again this is a DNA molecule, double-stranded, right? The area on the DNA molecule in green, so right here, is called the promoter. Okay, so it's just a special part of the DNA. It's got a special sequence of letters. And one of those sequences of letters or bases is called the Tata Box. And the reason it's called the Tata Box is because it has a bunch of T's and A's in it. So in this little dark green spot, there are going to be a bunch of T, A, T, A, T, A in a row. And that is a sign for a few other molecules called transcription factors to come in and bond in that area. And that's what these are. These little bubbles are other molecules that are attracted to that area of the DNA where there's all those A's and T's, and they stick onto it. And when that sticks onto that area, That is a signal for RNA polymerase, which is this kind of clear egg-looking thing here, to bond to that promoter region. So there's actually kind of a series of steps that has to happen for that RNA to find the beginning of the gene. Then once that RNA polymerase finds the beginning of that gene, then it can start making the messenger RNA there in red. The end of transcription is another area which signals the end of the gene called the terminator. And there are similar chemical components there that basically tell the RNA polymerase that this is the end of the gene and this is where you need to stop. Now, the mRNA, remember, carries the message of the DNA. But in order for the message of the DNA to mean anything, the messenger RNA has to leave the nucleus. The way it's formed right now, after it's been transcribed from the DNA, it is not able to leave the nucleus yet. There are a couple changes that have to occur in order for it to do that. So we're calling these changes that need to occur, RNA processing. And again, this also happens in the nucleus. The reason for RNA processing to occur, remember, is so that it can leave the nucleus. And secondly, so eventually this messenger RNA can attach to a ribosome out in the cytoplasm, which we'll talk about soon. All right, so without getting into too much detail, a couple of things that happen in order to get the mRNA out of the nucleus and get it attached to a ribosome. It gets what's called a 5' cap. You can see these are a bunch of phosphates. That's what the P's represent, with a guanine on it. But you just need to know, on the 5' end of the mRNA, put a little cap on there. And number 2, on the 3' end, get what's called a poly-A tail. Poly-A referring to the adenines. Many A's in a row stuck onto the end. And again, that's the 3' end. Okay? Once those two ends are taken care of, the mRNA can leave the nucleus. Now that it's able to leave the nucleus, we're going to pare it down a little bit so that it's not so many nucleotides long. Typical chunk of DNA for a gene, so one gene in DNA, is about 8,000 nucleotides long. After we go through this processing section, it'll get down to about 1,200 nucleotides in the RNA. So the processing cuts out quite a few of the bases. The way it does this is it takes scroll down a little bit here, it takes the RNA, so this is the RNA right here, and you see it's already got its poly-A tail, it's got its 5' cap, and it's going to have some enzymes that come and cut out specific parts of the mRNA called introns. Okay, so the introns get taken out of the messenger RNA. After those are taken out, the parts that are left behind, called the exons, so this section, and this section, reattach to form the actual RNA molecule that's going to leave the nucleus. Okay? One way you can remember it is the exons are the parts that are left behind to exit the nucleus. Okay? The reason they were called exons is because this is a part of the mRNA that actually expresses itself. But remember, the exons exit the nucleus with the mRNA. The introns are cut out. Okay? Introns are non-coding parts of the DNA. They don't carry any of the message. We're going to pare down the mRNA to just the basics of what the cell needs to know. All right, After that, the mRNA is able to leave the nucleus and go into the cytoplasm.